So welcome everyone to MTRI's April Sit-Back Seminar. My name is Chad Simmons and I'm an ecologist here at the Mersey-Tobiatic Research Institute. But before we begin, I would like to acknowledge that we are meeting here today and MTRI is based in Guestwick, Southwest Nova Scotia, one of the 7 districts of Mi'kmaq, homeland to the Mi'kmaq people. We want to acknowledge the treaties of peace and friendship and thank the Mi'kmaq people for their generosity in sharing their homeland with us. So for anyone unaware, MTRI is a science-based nonprofit, and we are nestled in Southwest Nova Scotia near Kejimkujik and within the Southwest Nova Scotia Biosphere Reserve. Our mission is to promote, conserve, and sustain biodiversity in Guestwick as well as beyond. So tonight, I'm very pleased to introduce Samantha Beal. Samantha is a Master of Science student at Dalhousie University, and tonight she'll be presenting developing environmental DNA into a biodiversity monitoring tool using the endangered Atlantic whitefish. So next, I can hand the seminar over to Samantha, but I'd like to remind everyone to please keep your mics muted. And if you have any questions during the talk, you can type them into the chat window if you're on Zoom. And if you're watching us over Facebook, you can type a comment and we'll get to them at the end of the presentation during the question period. So Sam, if you'd like to take over.
Yeah, well, thank you for the introduction, Chad. So like Chad said, I'm Sam. I'm currently doing my Master's of Biology. And today I'm going to be talking to you about eDNA and whitefish. So my talk today is going to be pretty informal, and I'm just going to be covering some of the background rationale for my master's research. I've structured it into 3 sections where I'll firstly be discussing why biodiversity is even something we should be monitoring. I'll then move into environmental DNA as a tool to do this, what exactly I even mean by environmental DNA, why there's growing interest in this technology, and how exactly it's implemented in a study. And then I'll finish up by talking about Atlantic whitefish as a model for this type of work. So biodiversity just refers to the variety of life on Earth. And this is variety not only between the different species, but also within the different species and within and between different ecosystems. And ecosystems and biodiversity are very important as biodiversity helps to support ecosystems and in turn supports the ecosystem processes, which provide us the goods and services which we rely on for our lives. So not only do they provide us our drinking water, but they also help to provide a livable climate and breathable air. In fact, some species actually have the ability to pull carbon dioxide out of the air and store it deep in the ground. Furthermore, I think we all know that a lot of people are employed based on environmental sectors, but it goes beyond that in that food crop diversity and the animals which pollinate these crops are very important, as if a disease strikes a food crop, diversity can help save the crop from total decimation, and we support our populations through agriculture. Our medicine is based off of plant and animal diversity. In fact, in the US, about 50% of all pharmaceuticals are made using natural compounds, many of which we cannot make artificially. And finally, there's also large cultural and intrinsic value which biodiversity and in turn whole ecosystems can provide to us. And this sounds great. We get a lot from the ecosystem, and we do. But unfortunately, the biodiversity which helps to sustain these ecosystems is in crisis. I would like to start this off though by saying extinction is natural. From the fossil record, we can see that the baseline extinction rate was about 1 species for every million species per year. And this extinction was typically balanced out by the evolution of new species. So when some disappeared, new ones could move in to fill the gap left behind. There were, however, 5 times in the Earth's history that saw elevated levels of extinction in a relatively short period of time, geologically speaking. And these events are characterized by having more than 75% species loss, which is kind of a mind-boggling number. Like that's 3/4 of species. And unfortunately, what we're seeing now is extinction rates are happening faster than even during those times. We can see on the graph here that records of extinctions really began to climb after the Industrial Revolution. And the current species extinction rates that we can see can be attributed to human actions, whether it's the fact that we are fragmenting and destroying critical habitat, or that we're inputting so much pollution into the environment. But we're also moving species around the globe, and this can cause some of them established in new regions that we're not previously in, and they can become invasive and decimate the natural populations. But also overharvesting and climate change are also leading to this biodiversity decline. And biodiversity decline will in turn have consequences for the ecosystem functioning which we humans rely on. So marine, terrestrial, and freshwater ecosystems are all experiencing biodiversity decline. But for this presentation, I will be focusing on the freshwater, as I am currently working on a freshwater fish. So these are small but mighty areas. They cover freshwater accounts for only about 3% of all the water on Earth, and yet over half of all fish that we have described are found within freshwater systems. And nearly 1 in 3 of these freshwater fish are currently threatened with extinction. We humans have high dependence on freshwater ecosystems. Not only do we get our drinking water from them, but they also help to support the livelihood of millions of people, oceans as well. About 5% of Nova Scotia's area is covered by freshwater systems, and there's over 6,000 lakes greater than 1 hectare in area within the province. And these lakes, like we're seeing everywhere, are not immune to biodiversity loss. So if you're thinking, okay, I need to go into a lake and see what's like this. So as you can see, there's a lot of nets, a lot of gear, and a lot of people. So not only can this be a very timely endeavor and therefore costly, because you not only have to pay for all the equipment, but also compensate people for the time it takes to deploy the nets and count the species, but it can also be costly to the fish in the terms that it's very stressful. If you imagine you're swimming in the ocean, you're swimming in a lake, and all of a sudden you're caught in a net, that's unfamiliar, that's stressful. Not only that, you then have to be handled as the researchers are going to identify you. They need to figure out what you are. And elevated levels of stress can actually cause harm and in some cases death to fish. So this brings me into the second part of my talk, environmental DNA, or as I'll be referring to it from here on out, eDNA. So what exactly do I mean by eDNA? Well, I have an analogy I would like to use. So bear with me. I think that most of us have probably heard that dead skin cells. Well, those skin cells, they came from you. They have your DNA. And fish do this too. They shed DNA away from themselves into the environment. And this can be in the form of skin flaking or mucus and saliva production. And just like the dust stays in our house even after we leave, the DNA can stay in the water body even after a fish has left the area. Our skin cells had to work their way off of us into the air before they finally sunk to the bottom and settled on the floor. Well, in the environment, eDNA also sinks, and therefore it can collect in the sediment as well. And it's collectible from both these environments. If you wanted to go out and do an eDNA study, there are 2 main types you could do. The first is where you're focused on just detecting a single species of interest. This could be a species that is commercially important or invasive, but also endangered. The second approach is a community-wide approach, and this is known as metabarcoding. And the goal with metabarcoding is to identify all species who have contributed DNA into your environmental sample. And this is possible because certain regions of the DNA are very similar across different species, but they're different enough that if you look at that region, you can use those differences to tell the species apart and figure out what they were. eDNA is actually a relatively new technology. The first paper to utilize it only came out in the late 2000s, and in this paper, the invasive American bullfrog DNA was detected from aquatic samples. Since then, interest in eDNA has only grown, with the majority of papers still focused on a single species approach. But as you can see with the light green bars, metabarcoding is growing as well. And metabarcoding in particular is a very powerful tool for assessing the biodiversity of a freshwater system, because if you sample over a long enough period of time, you could actually track changes in species distribution. So if you were thinking now you want to go out and do an eDNA survey, whether you wanted to do a single species approach or the metabarcoding approach, your workflow is going to be quite similar. Firstly, you need to actually collect your environmental sample. Here I've focused on water just because that's what the majority of eDNA studies still do. So once you've collected it, you now have DNA floating around in a water sample. So you need to have a way to isolate that DNA. And this is done by running the water through a filter. And the filter has pores, which are big enough for the water to get through, but small enough that the DNA can't. So now your DNA is on the filter, but it's still not at a part where it's workable. And so you need to extract the DNA off of the filter. Once you've extracted the DNA, you don't actually have that much to work with. And so you need to make many, many more copies. And this is done through PCR. And PCR utilizes primers, which are tiny strings of nucleic acid, which bind to specific pieces of DNA to allow for DNA replication. And this allows you to amplify only the DNA that you're interested in. And also PCR has really gained popularity in recent years because this is the technique that's used to diagnose COVID. And so once you've amplified and made millions of copies of your DNA, it's time to analyze it. There are many different methods to do this. The machine I have up here is an Illumina MiSeq, which actually is able to read the specific DNA sequence within your sample. And I've just highlighted it here because this is what we do in my lab. But as with everything, there are things to take into consideration when deciding if eDNA is the tool that's best suited for your research goals. So firstly, eDNA is fantastic because it is non-invasive. It does not harm the animals you're interested in because the animals don't even have to be present at the time of sampling. It has broad applications that can be used to answer many different research questions, and it can be applied in many different environments, whether that's a lake, a river, or the ocean. And also, especially when compared to the netting approach, it can be quicker and more cost-effective. But it's not without its drawbacks, and I don't want you to think it's a be-all end-all for everything. Namely, because the animal doesn't even have to be present at the time you're sampling, you can't gather physical information on it. So you don't know how big it fits into within the sample. Lastly, the DNA, once it gets into the environment, how long does it last before it gets broken up into pieces too small to detect? How far can the currents pull the DNA away from the original source location? And also how eDNA shedding rate varies not only between different species, but also different individuals within the same species. And so this brings us to the third section of my talk, where I'll be discussing my work with Atlantic whitefish. But I think it's important to understand them as a model species that we first need to discuss their history and how they gained their endangered status. So firstly, Atlantic whitefish are a member of the Salmonidae family, which means they're a relative of trout and salmon. And they're actually the oldest member of their lineage. Estimates for their divergence from the common whitefish ancestor are over 14 million years ago. So they've been around for a very long time. They have silver sides and a light belly with a dark back body. They're in the wild, they were reported as being between 20 to 40 centimeters in fork length, which is the length from the tip of the nose to where the tail fin forks. But some of the captive Atlantic whitefish have grown even bigger than that, with the biggest being around 50 centimeters in fork length. And they're quite similar to lake whitefish, their relative who is more widespread throughout Nova Scotia. But physically, there are some differences. Namely, Atlantic whitefish have more teeth and more vertebrae, and their mouth points forward, where lake whitefish mouth points more downward. And I highlight the differences because I think it's important to be able to identify them, as fishing for Atlantic whitefish is actually prohibited due to their endangered status. So the habitat requirements for Atlantic whitefish are not that well defined, and there's a lot of unknowns around them. However, this is a species who historically would migrate from the freshwater, from the ocean up through the freshwater system that is accessible to both the marine and freshwater environments. Fish surveys conducted across the province between the 60s and the 80s only found Atlantic whitefish in two regions, the Tusket River down near Yarmouth County and the Petite Rivière south of the town of Bridgewater. But unfortunately, the Tusket River population no longer exists. Atlantic whitefish have not been documented there since the early 1980s. And there are many factors which are thought to have contributed to their disappearance, namely overfishing, the water quality becoming too poor due to acidification, blockage of fish passage between the marine and freshwater environments, as well as the introduction of invasive species. And so this disappearance of the Tusket River population represents a 50% decline in Atlantic whitefish range in only a few decades. If we zoom into the Petite Rivière, this is an area that is covered with forests and is categorized by the presence of its many bogs. And it actually provides Bridgewater with their drinking water. And so the region underwent damming in the 1800s. And what's pretty amazing is that Atlantic whitefish were able to adapt, and they now perform their whole life cycle within these freshwater lakes. Although the watershed comprises the entire yellow area on the map, Atlantic whitefish have only been documented in the middle 3 interconnected lakes. And together, these lakes span 16 square kilometers in area. They are relatively shallow, with the deepest, Menamkeek, which is the most south lake on the map, reaching only 16 meters in certain parts. And when compared to other watersheds along the south shore, these lakes are on the acidic side. Although Atlantic whitefish are found in the 3 lakes, they do comprise just a single genetic population, which means there's no real genetic difference between a fish found in one lake versus another. So the threats that Atlantic whitefish are facing in the Petite Rivière are very similar to those which impacted the Tusket River population, namely the barrier to fish passage in the form of damming. And although Atlantic whitefish were able to adapt and persist in the freshwater environment, the dams meant that any fish who washed over the dam had no way back into the lake and was therefore completely removed from the reproductive pool. The Petite is also undergoing habitat alteration in the form of acidification. And acidification can impact the ability of a fish to actually pull oxygen out of the water. And they're just like us, they need to breathe to live. And so if they're struggling to pull oxygen out of the water, that can be very detrimental. But it's not just historical threats. Just in the last couple of months, there's been proposed logging on the Petite Rivière, right up to the shore of Menamkeek Lake. And logging can have really bad consequences, as it can lead to soil erosion and the input of more sediment into the water body. And this can in turn affect the quality of the water, and it can also change the bottom habitat of the lake, which may have implications for spawning. Further, logging can cause a reduction in tree canopy, which is the amount of leaves over the lake, and this can impact the water quality and oxygen levels, which can have consequences for the fish within the lake. Aquatic invasive species have also worked their way into this system. Smallmouth bass are a competitor for Atlantic whitefish, as they both go after the same type of food. However, chain pickerel can and do eat Atlantic whitefish. They are a voracious predator who can eat them. I would like to walk you through some of the previous recovery efforts that have gone into Atlantic whitefish, because I think it's important to acknowledge that a lot of work has already been done on the species long before I started my thesis. When the Committee on the Status of Wildlife in Canada was formed in the 80s, Atlantic whitefish were listed as endangered. And it was only about a decade later that smallmouth bass were found in the system. In 2000, there was the Mersey Biodiversity Facility. And although it was not created with the ultimate goal of Atlantic whitefish, it did end up running a successful captive breeding program for the species. When the Species at Risk Act was implemented in the early 2000s, Atlantic whitefish were again listed as endangered. And so between these two listings, they were granted legal protection. The captive breeding program did release juvenile Atlantic whitefish, not only into the Petite Rivière, into Heb Lake, but also into Anderson Lake, which is a small lake north of Dartmouth, indicated here on the map with the red pin. And this was done with the hope of establishing a new population. But unfortunately, when they went back to assess it, they did not detect Atlantic whitefish. And so this effort was ultimately unsuccessful. 2012 saw ocean passage restored at Heb Lake when a fish ladder was implemented at the dam. And this ladder meant that now any fish who washed over the dam had a way to rejoin the population and stay within the reproductive pool. But unfortunately, this is also the same year that the captive breeding facility not only was closed.
If you want me to do
I
it.
get physio and acupuncture and
Sorry.
Sorry about that.
Oh, no worries. And then it was only a year later that chain pickerel were detected in the Petite. And once chain pickerel are detected, things really start to ramp up. So 2014 saw DFO began electrofishing in Heb Lake to remove the invasive species. The next year, rotary screw traps were placed at the connections between the three lakes, and this had two goals. Firstly, it was to catch any invasive species who happened to be moving between the lakes, but it was also done to monitor Atlantic whitefish activity, because even if adults are hard to find, if you're finding larvae, that means adults have survived at least long enough to reproduce. In 2018, though, the larvae were not only counted, but they were collected and brought to Dalhousie to be raised in captivity away from the predator-ridden lakes. And 2018 was also the year that electrofishing was expanded beyond Heb to the three lakes of the system. And it's very exciting that just this year, there was successful reproduction with the Atlantic whitefish at Dalhousie. So Atlantic whitefish are a model species for eDNA work because there's already ongoing interest in their recovery. There are existing recovery strategies, and so we can implement eDNA alongside more traditional methods. They are a very elusive species and difficult to find, and so the non-invasiveness of eDNA really offers a great opportunity to try to detect them in the wild. And lastly, because they're directly threatened by two other fish species, we have the opportunity to develop and try both the single species approach, but also the community approach. So my research has four main goals, where I'm firstly trying to locate them in the wild, but I'm also trying to determine when and where they're spawning in these lakes. I'm curious to see if the amount of aquatic invasive species DNA differs before and after the electrofishing events. And lastly, I would like to assess the community makeup of the Petite Rivière by using metabarcoding. So the first step of my research is to develop an eDNA workflow. So coming back to this picture I showed earlier, there are a lot of variables a researcher needs to decide on at each step in the process. Firstly, you need to decide how much water do you want to collect and where do you want to collect it, but also how. You then need to decide on a filter type and an extraction method. You need to decide what primers do you want to use to amplify the DNA. And then you also need to figure out how to analyze the DNA data once you have it. So once the sampling plan was in place, it was time to actually go to the water and collect the water samples. So this was done by sampling both just under the surface, but also at depth, because Atlantic whitefish are thought to occupy the entire water body. And so we just wanted to try sampling at different locations to see what we found. The next step was to sample at different points in the lake at different times during the year. So the red dots on this map show where electrofishing occurs. And we collected water samples about 1 week before electrofishing happened and about 1 week after it finished. And this sampling was in the late summer through to the early fall. The green dots represent sampling during the spring larval emergence. And so samples were taken when Atlantic whitefish larvae were present in the rotary screw trap. And the goal of this was to see if we were able to even detect an eDNA signal from a larva, because they are quite small, so we didn't know how much DNA they would be putting out there. And the blue dots represent trying to locate spawning ground. So these samples were chosen because even though we don't know a lot about Atlantic whitefish preferences in the wild, we utilize knowledge on their relative lake whitefish to pick areas which would be suitable for them as a starting point for Atlantic whitefish. So that was how we decided on the location. But in order to determine the timing of spawning, we conducted the sampling once a week for 8 weeks, with the thought that because Atlantic whitefish are broadcast spawners, which means that they release their eggs and sperm directly into the water column, that would be a big influx of genetic material. And genetic material contains DNA, which we should be able to detect from the water sample. And really the goal of this is to identify critical regions in the lake for concentrated recovery efforts at the most important time of the year. And now I hope I haven't painted a bleak picture of Atlantic whitefish, because there are a lot of reasons to hope for this species. Firstly, in 2018, the not range beyond the Petite, as well as restoring migration between the ocean and freshwater. And what's amazing is that even though these fish have been landlocked for over a century, they still do retain the ability to tolerate salt water, as was determined through previous experiments. I also would like to highlight that the successful reproduction at Dalhousie's aquatron is no small feat. It has been over a decade since the last it's been about a decade since the last captive breeding program closed, and yet they did it again. And so as you can see here, there's maybe hundreds of little Atlantic whitefish. And so these can help to meet the first two goals of the recovery plan. And lastly, I walked you through that very long timeline because I think it's important to acknowledge that we can learn a lot from the past, even if those recovery efforts at the time were ultimately not successful. We can take what was learned and apply it to the future. So if you're wondering, there are some things that you yourself can be doing for Atlantic whitefish. Firstly, I think it's important to learn how to identify them so you can release them if you accidentally catch them if you're out fishing on the Petite. Secondly, if you do happen to see one, please report it to the fisheries departments. This is a big one, but do not move fish between the different lakes. It's really even bait species. And lastly, just talk about Atlantic whitefish. Talk about it with your community. Tell your local government that you support protecting them and their habitat. This is a fish that is only found in Nova Scotia, and it's ours to protect and ours to be proud of. So the significance of this really comes down to the fact that Canada is not exactly known for its biodiversity, and yet Nova Scotia is home to a fish found nowhere else in the entire world. They are one of only 4 freshwater fish only found in Canada. Furthermore, they are the oldest member of their whitefish lineage, and so they really represent an evolutionary significant part of our Canadian biodiversity. And yet the risk of extinction is there. Lastly, the framework set forth in this work can then be applied to other at-risk freshwater species. So to summarize, biodiversity is important, yet it is declining across the board, and freshwater ecosystems in particular are at risk. Atlantic whitefish are one such freshwater fish that is currently endangered, and eDNA is a promising method to detect them in their natural habitat. So as with everything, it takes a village. So I would like to thank my supervisors, lab manager Ian and research associate Beth, as well as everyone else in the Benson lab, John Batt and the Aquatron staff, and everyone who's working really hard to keep the captive whitefish thriving and reproducing. And then as well as everyone who has collected water samples from me and who is working towards the conservation of this species. So, and I'm happy to take any questions now.
Great. Thank you very much, Sam, for that excellent, sobering talk, but also very optimistic. I love it. So we do have a question in the chat, and Anna asks if you can explain electrofishing a little bit more.
Yeah, so electrofishing, sorry, I should have included a photo, but basically they go out on a boat and they have a rod that's attached, it's an electric rod. And so they put it on the surface and it shocks just a little bit of electricity into the water, not enough to kill big fish or anything. And so mainly this is done to get the juvenile aquatic invasive species. And so once they've stunned the fish, they can net them and take them out of the habitat.
Great. Does anyone else have any questions? Well, I have some questions.
Okay.
So do we have a good idea of how climate change is going to affect the Atlantic whitefish?
So I am not particularly sure about that, but I think it's safe to say it will affect them if water temperatures rise and they actually do seem to prefer cold water. In the summer when the lake stratifies, they're thought to actually go beneath the thermocline and stay in the cold water. So we're not sure how warmer water would affect them, but it's safe to say that it probably might cause some problems.
Great. And just another question. Oh, I see Shannon has a question.
Hi, yeah, thank you. And thanks, Sam. This was a great talk. I'm super excited to hear that eDNA work is going on and that you guys are finding the whitefish. So my question is, so I know this is a fairly new science and that you guys are establishing the framework and learning how to do this. But I was wondering, in the future, do you foresee like a way for the community and people who live around those lakes to participate in the eDNA monitoring in a way?
Yeah, that's an interesting one, and I would love it if it was something that could become a citizen science program. But unfortunately, the issue with eDNA is it's pretty sensitive. And so you just have to make sure on the sample be contaminated. And there's also the issue of once you collect the water, you have to filter it within 24 hours because the DNA will degrade in the bottle. So I think there's also a lot of logistical problems that would come up in trying to do any sort of large scale community eDNA monitoring. But that's not to say the community can't be involved. I think most important is just talking about this species, because I mean, I don't think they get the PR that they deserve.
Thanks.
Yeah.
I'm just a big fan of this type of science, so
Oh,
I'm wondering if people can participate.
Yeah, I mean, maybe in the future, I know there's some work being developed about like filtering on site, but I don't have any experience with that, so I can't really speak to it.
Okay, interesting. Thank you.
Yeah, no worries.
I just have a bit of a follow-up. So you said like the DNA starts to break down within like 24 hours once you collect it. Is that generally how long it lasts in the environment before it's too degraded?
So that's what we're really not sure. And a lot of more work does have to be done to figure out exactly the degradation time. The 24 hours, I think, is just a good buffer to be sure. But yeah, and especially in the water column, at least, the degradation can be impacted by the temperature, how much sunlight it's getting, and the microbial community at the time. So it really is ecosystem dependent.
Great. So Lori asks, you might have said this, but is the primer for Atlantic whitefish developed?
Yes, so my lab has been doing a lot of work to develop the primers because we're playing around with targeting a couple of different types of DNA in different sections of the DNA. But yeah, so we do have working primers.
Great. And Lori also asks, what do you think will be the biggest challenge of your project?
I think the biggest challenge is that there's not a lot of Atlantic whitefish in the wild. We don't actually know how many are there. And so 16 square kilometers doesn't sound like a lot until you go out and collect, 1 liter at a time. And then you see just how big 16 square kilometers really is. So I think the low numbers definitely poses a challenge, and the fact that not a lot is known about Atlantic whitefish in the wild. So it's even harder to try to find areas that we think would have high probability.
So Marion asks via our Facebook Live, are you seeing any issues with goldfish and them being an invasive species?
Within the system, I'm not sure actually. I haven't really dug into the data yet. I'm still just in my first year. So that would be definitely something to be aware of, is all the other species. I just highlight those invasive species because they're known to directly threaten Atlantic whitefish.
Okay, does anyone else have any questions? No? So Amy says, no goldfish thus far in the upper Petite, only smallmouth bass and chain pickerel have been detected. Thank you, Amy.
That's good to know.
Great. Okay. So if there's no more questions, thank you again, Sam, for that really incredible talk. And thank you everyone for joining us tonight. You can also join us next month on the last Thursday of the month, which is May 26th, for our next Sit Back seminar, when we will be joined by Hannah Sewell, also from Dalhousie University, for their presentation on how humans are impacting baleen whale distributions. So with that, I would like to thank the Region of Queens for supporting us here tonight and our seminar series. And as always, you can stay up to date with MTRI seminars by following us on Facebook. And if you'd like to rewatch tonight's seminar again, you can check us out on Facebook or our YouTube channel. So with that, we hope everyone stays well and we get to see you all again very soon. Thank you, everyone.
Yeah, thanks for coming.